Hello and welcome to my channel. In this lecture we'll continue our talk about the nervous system, so let's start. After studying the brain, now it's time to study the coverings of the brain. The dura matter and the arachnoid matter and the pia matter. The first layer is the dura matter, the one you see here. It's composed of two layers. Periosteal layer, it's the endosteum of the skull actually, that lines the skull and continues through the foramen magnum to the vertebral column. So it's adherent to the bone. While the meningeal layer is the one visible here, is the one that covers the brain. It's a thick membrane and it's in close contact with the arachnoid and continues through the foramen magnum as the spinal dura. It forms dural partitions like the falx cerebri, the falx cerebelli, Tintorium cerebelli, and diaphragma celli. Two of these are vertical, the falx cerebri and the falx cerebelli, and two of these are transverse or horizontal, which are the Tintorium cerebelli and diaphragma celli. The falx cerebri is the one you see here. It's broader posteriorly than anterior. Anterior, it's attached to the crista galli, a protrusion from the roof of the nose. Of course from the cranial bones, and attaches itself posteriorly in the confluence in the internal occipital protuberance and continues laterally in both sides as the Tintorium cerebelli. It attaches to the roof of the Tintorium cerebelli, as you can see here. It has the superior sagittal sinus against the skull, and in its free border has the inferior sagittal sinus. At its base, it has the straight sinus formed by the great cerebral vein of Galen and the inferior sagittal sinus joining together. Tintorium cerebelli, this is half of the Tintorium cerebelli, this half is removed. It's a transverse fold attached to the petrous bone and the internal nuchal line, or the transverse process of the skull from the inside internal occipital protuberance to the transverse process. As along its attachment runs the transverse sinus, as you can see here. It has anteriorly, it attaches to the anterior and posterior clinoid processes, and in the middle it has a free border which forms together with the other side the tentorial notch for the brainstem to pass through. Sometimes, by the way, the brainstem herniates through this notch into the foramen magnum if increased intracranial pressure, for example, by a tumor. The falx cerebelli is this thin vertical fold, runs between the two cerebellar hemispheres, and it's small, attaches posteriorly to the internal occipital crest. Diaphragma celli is another horizontal fold, the one you see here, that covers the pituitary gland, but pierced in the middle to allow for the passage of the infundibulum that connects the pituitary gland to the hypothalamus. This is the diaphragma celli, covering completely what's so-called the sella turcica between the clinoid processes, the depression between the clinoid processes. The arachnoid matter is thin avascular membrane. It covers the brain but doesn't go through the sulci, except in the interhemispheric fissure. It has internal extensions that connect to the pia mater, underlying pia mater, through the subarachnoid space, and at the superior sagittal sinus it has projections called the arachnoid granulations, essential for the drainage of the CSF. Continues, of course, with the dura through the foramen magnum as the arachnoid mater of the spinal cord. Pia mater is as a layer of endothelium that covers the brain, runs through the sulci and gyri, and it's a delicate membrane, and it differs from the other coverings that it runs through the sulci and gyri of the brain, closely adherent to it, as you can see here. There are spaces formed by the coverings of the brain. The space above the dura, between it and the skull, is called the extradual space. And if you perform an X-ray, for example, and found a hemorrhage in this area in the extradual space, know that the artery responsible for that is the middle meningeal artery, which runs in the space. Subdural space between it and the arachnoid is a potential space filled with blood in the X-ray in the case of cerebral veins injury, because the cerebral veins traverse the space, the subdural space, to supply the brain. Subarachnoid space between the arachnoid and pia, it's filled with CSF and has the blood vessels of the brain running into it. It enlarges to form cisterns, means collections of CSF, forms the choroid plexus in the ventricles, and has the choroid plexus in the ventricles, and the choroid plexus are responsible for production of CSF. The arachnoid villi mentioned before, responsible for the drainage of CSF into the corresponding dural venous sinuses. The ventricular system, its cavities lying within the concavity, or within the substance of the brain, composed of two lateral ventricles connected through underneath the fornix through the interventricular foramen of Monro to the third ventricle, and then from the third ventricle through the aqueduct of Sylvius to the fourth ventricle, as you can see here between the pons and the cerebellum, and through a central foramen and two lateral foramina, the CSF encloses the spinal cord that will form in the vertebral canal. The CSF is about 150 milliliter of watery fluid. It has 4 times turnover a day, means that 500 milliliters formed every day, and forms cisterna, as mentioned before. Examples of such cistern: interpeduncular cistern, the supracerebellar cistern, the cisterna magna between the cerebellum and the medulla, the supracallosal cistern. All are collections of CSF because of the potential spaces formed by the arachnoid. The ventricles are derived from the inner lumen of the developing neural tube in embryology, and they adapt to the shape of the surrounding walls. As you can see here, we have the lateral ventricle, which has an anterior horn, posterior horn, and an inferior horn. Anterior horn, inferior horn, posterior horn, and this is this area of beating called the body of the lateral ventricle. As you can see there, the two lateral ventricles communicate together through the interventricular foramen of Monroe to fill the third ventricle between the two thalami, then through the aqueduct of Sylvius to the fourth ventricle. The third ventricle lies in this area between the two thalami and traversed by the interthalamic adhesions and via the aqueduct of Sylvius between the tectum of the midbrain and the tegmentum of the midbrain communicates with the fourth ventricle between the cerebellum and the pons. It has some recesses like the infundibular recess, the optic recess, the pineal recess. Means extensions in the nearby structures. Fourth ventricle, this is a posterior view of the brainstem, cerebellum removed, of course, to visualize the posterior surface of the brainstem. This is the midbrain, and this is the pons, and this is the medulla. This is considered the floor of the fourth ventricle. You can find many structures like the median eminence, for example, the facial colliculus, the trigonal, hypoglossal trigone, and the vagal trigone. It connects through the foramen of Magendie to the central canal of the spinal cord and through the lateral foramina of Luschka to the sides of the spinal cord. So now you have a rough idea about the circulation of the CSF. CSF cycle, it's formed by the choroid plexus in the ventricles. As you can see here, examples of choroid plexus. They are composed of blood vessels covered by special endothelium, the choroid epithelium, formed of cells that are tied together to form the blood CSF barrier, not the blood-brain barrier. Don't get confused with that. We'll talk about this later. It's formed to fill the lateral ventricles, then the third ventricle, then through the aqueduct to the fourth ventricle, then around the spinal cord and around the brain, as you can see here. So you can consider the brain floating in CSF because it's filled with CSF from the inside and surrounded by CSF from the outside. By the way, the brain weighs around 1.5 kilograms to 2.5 kilograms. But if you, in situ, if you put an electronic weight measure on the base of the skull, you will find that that the weight of the brain averages 200 milligrams. See, it's a great difference in weight. So this arrangement, the floating process, allows the brain to maintain its shape because it's gelatinous in consistency. If you extract it and put it on the table, for example, it will collapse. This is essential for the function of the cranial nerves arriving, or the structures arising from the brain. Drainage of the CSF by means of the arachnoid villi, the ones you see here, draining into the superior sagittal sinus. Impaired drainage is due to blockage by RBCs, examples in hemorrhage, for example, or white BCs, or or proteins in infections or fibrosis after as a complication of meningitis. They may be few in number in babies, and tumors above the tentorium prevents upward flow of CSF through the tentorial notch, leading to increase in pressure below the Tintorium Cerebelli. The blood CSF barrier we talked about, it's a barrier between the blood and the CSF, present at the choroid plexus. Because there are tight junctions between the choroid epithelial cells, and please do not confuse that with the blood-brain barrier, which is formed by the foot processes of the astrocytes on the surface of brain capillaries. Blood-brain barrier, this is the blood-brain barrier formed by the lining endothelium of capillaries, the basement membrane of these capillaries, and the foot processes of the covering astrocytes. It's present everywhere in the brain except in the choroid plexus, the hypothalamus, the pineal gland, area postrema, and the medulla, for the function of these areas, because they measure osmolarity and glucose. So they must have direct contact with the blood. Structures passing through this barrier are lipid-soluble structures like alcohol and anesthetics, CO2, O2, and H2O. And it's slightly permeable to sodium, chloride, and potassium. It's impermeable to plasma proteins, which have big molecules, and non-lipid soluble large molecules. So for the drug, for example, to act centrally, if a drug is said to act centrally, means that it acts directly on the brain, it must has the ability to cross the blood-brain barrier for its function. Hydrocephalus, as you can see here, means excess water in the cranial cavity. We have two types of hydrocephalus, communicating type and non-communicating type. Communicating type is due to blockage at the arachnoid villi and fluid increase inside and outside. The result is a huge increase in size of the skull, especially during infancy, because you know the presence of fontanelles still. Signs of which we have the sunset sign, as you can see here. The eye, the pupil is pushed down, the failure of closure of fontanelles. Non-communicating type of the hydrocephalus is due to atresia or blockage of the aqueduct of Sylvius, either congenital or by tumors. Atresia means narrowing. The pressure increases in lateral and third ventricles, and the treatment is by silicone rubber tube connecting the lateral ventricle and peritoneal cavity. Of course, it has to be changed every now and then to allow for the growth of the child. Functions of CSF: because it lines the brain and covers the brain from the outside, so it has a cushioning action. It has the same specific gravity of plasma, so the brain floats in it. We mentioned that before. For the brain to maintain its shape, it's essential for the function of the brain. In trauma, because the brain is floating, so it has inertia. Boxers have what's so-called the counter-coup injuries, means the front trauma lead to posterior injury because of the inertia of the brain. So it hits against the inside of the skull anteriorly, while making a tremendous tension on the posterior surface of the brain. That's why the injury results on the posterior aspect of the brain, not the anterior, not at the size of trauma. And the opposite is correct. If the trauma is from the posterior side, the injury will be at the anterior side. It provides sun nutrition for the brain and acts like a sink, washes unwanted materials, and allows for movement for substances from the brain to the blood. I hope you got the maximum benefit from this lecture, and thank you very much for watching.